All right, let's talk about one of the most critical steps in medicine. How do you figure out the very first safe dose of a new drug to give to a person? It's this incredible scientific bridge between a lab animal and a patient. And getting it right is everything.
I mean, really think about that for a second. This is one of the highest stakes questions you can ask. You might have a drug that works wonders in a mouse, but how on earth do you translate that success to a human being safely? And the pressure is immense. Because get this, over 92% of drugs that make it to clinical trials ultimately fail to get approved. So getting that first dose right isn't just about safety. It's about giving a potentially life-saving medicine its absolute best shot at working. Okay, so let's dive into what scientists call the first-in-human problem. It's all about this challenge, right? Bridging that massive biological gap between a test species and you.
So what they're looking for is a reliable starting point. It's called the human equivalent dose, or HED. You can think of it as the ultimate sweet spot, a carefully calculated dose that's just strong enough to potentially work, but well within the limits of being safe.
For decades, the way we've answered this question has been through a set of really clever, standardized mathematical rules. This has been the foundational rulebook for drug developers for a long, long time. The most common method, the one that's basically a regulatory standard, is called body surface area scaling, or BSA. The big idea here is that a lot of biological stuff, like how a drug gets processed, scales better with surface area than with just plain old weight. It's a much smarter way to compare, say, a tiny mouse to a full-grown person. And the formula itself is actually pretty straightforward. It boils down to 3 steps. You start with what you know as a safe dose in an animal, then you use this special conversion number called a Km factor, and then boom, you use a simple ratio to calculate your starting dose for a human. Now, BSA scaling isn't the only classic method. Scientists have a whole toolkit. There's allometric scaling, methods that use what's called the no observed adverse effect level, and sometimes they just apply a huge 100-fold safety margin just to be cautious. For years, this was the standard approach because, honestly, it's pretty simple. But, and this is a really big but, biology is messy. It's incredibly complex. And these simple rules, as useful as they've been, have some major limitations. They can lead to getting the dose wrong, either too low so the drug looks useless, or worse, too high, which can be incredibly dangerous. See, the really crucial point is that these simple conversions are basically blind to what's happening at the biochemical level. They just can't see the complex machinery, all the unique enzymes and transporters that are totally different in a rat versus a human. This is a huge problem for certain drugs, especially ones that target the brain, where the differences between species are just enormous. And this slide just lays it out perfectly. It shows the massive philosophical divide between the old way and the new. We're moving away from what's basically a simple conversion chart and toward what you can only describe as a flight simulator for a drug. So let's talk about that flight simulator. This is the modern solution, building a virtual patient, a computer simulation, to test drugs on before they ever get near a real person. This incredible approach is called PBPK modeling, and it is a massive leap forward. Instead of relying on a simple formula, scientists build a super detailed digital twin of human physiology. This model can then predict exactly how a drug is going to be absorbed, distributed, and cleared from the body. But to build one of these virtual humans, you need a mountain of data. We're talking everything from the blood flow rates in different organs and the chemical makeup of tissues, all the way to the specific activity levels of key enzymes in the body. It's incredibly data intensive. And that's why this flight simulator analogy is just so perfect. It lets researchers run thousands of what-if scenarios in complete safety. What if the patient is older? What if they have kidney problems? You can test it all in the computer, long before the first real flight in a human volunteer. And PBPK isn't the only advanced tool in the box. You also have sophisticated statistical methods, like Bayesian models. These are great because they can pull together data from lots of different animal studies, and, crucially, they tell you how confident you can be in the final prediction. So, what does this all mean for the future of medicine? Well, it means we're entering a new era with a much, much smarter and more expansive toolkit for keeping patients safe. And what's really important to get is that these new fancy models don't just throw out the old rules, not at all.
Instead, they create this powerful integrated system. A scientist might use a classic scaling rule to get a ballpark number and then use a complex PBK model to really refine and validate it. And looking down the road, wow, this journey is far from over. Right now, the focus is on making today's models even better. But in the next decade, we're going to start feeding them data from incredible technologies like organ on a ship. And further out, we're talking about using AI and machine learning to predict the perfect dose for your unique individual biology. Which leaves us with one last pretty provocative thought. As these virtual models get better and better, so good that they're basically indistinguishable from reality, will animal testing, a cornerstone of drug development for centuries, finally become a thing of the past? It's a huge question that could totally change the future of medicine.